Okay, well, I think I'll go ahead and start with our introduction. I'd like to thank Peter Kalivas very much for joining us today from South Carolina. One second. For those of you who are new to PARC, PARC is the Pain and Addiction Research Center at UCSF. It was launched just over a year ago with a pretty wide variety of emphases to connect us across our campuses, across types of research, and across disciplines, including facilitating the development and testing of new approaches for treating pain and addiction, as well as improving our understanding of the underlying biology of these disorders and decreasing the negative consequences of opioid use. I invite you to join PARC. You can either scan our little QR code here or just search for PARC UCSF in your favorite search engine and sign up to join the organization and get announcements about these talks. And this is a photo of our retreat in person from last year, about a year ago now. And please consider joining us for future events. Oops, let me do this. So Peter Kalivas has been doing research about addiction and substance use disorder for quite a long time now. He's had over 400 publications and many awards in this area. And his research is really focused on characterizing the molecular neuroadaptations that occur in substance use disorder, as well as determining the critical neural circuitry that underlies behaviors related to addiction, with a real clear focus on relapse behaviors, which is really critical, I think, as a target for when we might be able to treat people with substance use disorder and help them to have fewer relapse events. Peter earned his PhD in pharmacology at the University of Washington, completed postdoctoral training at the University of North Carolina. He started his first assistant professor position at Louisiana State in New Orleans, and then quickly moved back to the West Coast to Washington State. And over the years, climbed in the ranks there and was the director of their Alcoholism and Drug Abuse Institute. In 1998, he moved to MUSC, where he is now, and is chair of the Department of Neurosciences in the College of Medicine. And his work has been really seminal in quite a few of the aspects of our understanding about drug abuse and the molecular and circuitry underpinnings of the behaviors involved. So thank you again, Peter, for joining us, and let me give the slideshow over to you.
Thanks, Melissa. I appreciate the introduction. Thanks for the invitation. It's really a pleasure to be here and share with you some of our recent data. Everybody can hear me fine? We're all good? Okay, so the title's a little different. I couldn't remember exactly what title I gave you, so this is just not too far off. Many of you, not many
Peter,
of you, but
Peter,
people.
I think you need to share.
Oh, is it not sharing?
I don't see them.
Oh, well, then I do need to share, don't I? Yeah, sorry about that. Okay.
That's great.
So there's the title. It's very close to what you had up there. And people from the West Coast always ask me how a guy who grew up in LA could end up in South Carolina. And so this was just a couple mornings ago when I woke up. This is a sunrise across the marsh in my backyard. It's a state of really incredible natural beauty if you like beaches and inland marshes and things like that. You're from San Francisco, so you probably do understand why I would leave Los Angeles, but that's a different story altogether. Okay, we're going to talk about how the neurobiology of how thoughts about drugs intrude and lead to relapse. I understand this is a translational group, so I've set up. Let me see why we're not advancing. I'm sorry, don't seem to be advancing here. There we go. Okay, so there's 4 parts of the seminar, and I've tried to weave in some of our clinical work. I'm not the clinician, I collaborate with clinicians, but I will frame the question in a more clinical way, and then go into the biology, and then come back out to a clinical trial. So first part of the talk is thoughts about intrusive thinking as a transdiagnostic phenotype in neuropsychiatry. And I'll explain that in a minute. I'll give you the latest things that we're doing in what we call the tetrapartite synapse and the plasticity. And this, try to introduce you if you're not already familiar with it. The general idea that you can't really understand synaptic plasticity unless you consider not just the pre and post synapse, but the extracellular matrix and the near adjacent astroglia. Then I'll jump into the ensemble hypothesis of brain circuits and how this is a nice transition between the more cell biology that we're going to get in part 2 and kind of bring it back out a little bit into the circuitry level of understanding and some of the things we're doing recently and how the hopes we have for those recent experiments. And then some clinical trials that are based on discoveries made mostly in number 2 there, and the results of those and where we think they're going. Because we haven't given up yet, even though it didn't work so well in stopping relapse, we're getting a very strong signal in PTSD, and that may not sound like that should happen, but I'll try to explain to you why we think it is happening. So how do thoughts intrude? I think to understand that, first let's get a definition of spontaneous thinking. So this is just a recent definition that was in a review: thoughts that unfold effortlessly and unrestrictedly. So these are things that we've all experienced: mind wandering, daydreaming, creativity, straight up dreaming when you're asleep. We spend about up to 50% of our waking life is spontaneously thinking. So we're just kind of going between one association and another. The more we're paying attention, the tighter those associations are with each other until they become very deliberate thinking, such as planning how to respond to something that's motivationally relevant. That would be very deliberate. It would no longer really be spontaneous thinking. We have about, if you're just floating along, doing your daydreaming, you'll have a transition about every 15 seconds. So this is a very normal process. The current thinking, as I understand it, is that it's probably facilitating memory consolidation and learning associations between things that you've already consolidated. So what's happened when this becomes intrusive is it's a form of very deliberate thinking. In other words, it's very structured thinking. You're paying a lot of attention to it, but it's not adaptive. So any distinct identifiable cognitive event that is unwanted, unintended, and recurrent interrupts the flow of thought, interferes with task performance, is associated with negative affect, and is difficult to control. So that covers a lot of ground, but it also, those of you that are psychiatrists, you can probably recognize that you're starting to this broad enough definition that it kind of fits in with symptoms of a lot of psychiatric disorders, and we'll come back to that. So, as I mentioned, I'm a neurobiologist. Most of my work is not with people, but it's with rodents. And with rodents, of all these things, the easiest thing to quantify and some would argue the only thing you can really quantify, is task performance. And so we're gonna, well, the biology I describe will be all about performing a task and neurobiology that's associated with that. And then that we think is linked to what happens in intrusive thinking. Okay, so let's just give an example of intrusive thinking, maybe something that some of you might have experienced at some point. So I'm here at work, get a call from a buddy who's just flown into Charleston and says, hey, we're all getting together at at this bar, and why don't you come down and join us? And that sounds like a pretty good plan to me. So I hop in my car and start driving to go to the bar to get a beer. And on the way, Sue calls me and says, hey, I'm going out with some friends, and you're going to have to come home and make dinner and do the homework with the kids tonight. So that's also very motivationally relevant. And that has its own plan. I would drive home and cook dinner, help with homework, etc. So this is now I'm faced with this decision. If I actually suffer from substance use disorder, what would happen is I would ruminate on what's going to happen at the bar. I'd be thinking about how fun it's going to be. I would start getting all excited about the bar, and the instructions I'd gotten from Sue would slowly fade into the background. Those of you that know Sue would know that I would go home. But if I was suffering from substance use disorder, I would end up doing this. This would come to dominate my motivational state, and I would head off to the bar. I might have a rationalization like I'll just have one beer and then go home, something like that. But the bottom line is I would relapse. So this relapse event is two components. You're getting the beer, the actual using of the drug is one component, but the component that we're really interested in, this intrusive component that starts to dominate my motivation to action circuitry, is really what we call craving. Or if you're in a laboratory with people and you're showing them cues that they would associate with the drug, they call it cue reactivity. Basically the same thing. So this is something that exists. If you call it intrusive thinking, because craving is indeed an intrusive event, if you call it intrusive thinking, you're basically dealing with something that is shared with other psychiatric disorders. So intrusive thinking is very important in post-traumatic stress disorder, where a person's thinking about their combat experience. They get a cue, something in the environment that reminds them of the experience. It comes back to them, and they can't regulate the anxiety that's associated with that experience. And so that intrusion is actually something that often can precipitate a state of high anxiety that we would associate with PTSD. Rumination and depression, or I'm assuming chronic pain. I haven't thought about that one as much, but I thought about it a little bit in putting this slide together this morning. That maybe chronic pain, there's a lot of rumination that takes place on that as well, I think. So there could be a symptom of chronic pain, could be intrusive thinking, and OCD is kind of an obvious one, where the movement disorders many times, or thought process, intrude and disrupt your ongoing adaptive motor behavior. Okay, so this is really what we're going to study, and I'll talk about it mostly in terms of craving and addiction as an animal model. And then in the clinical trial, we'll slip into PTSD. I don't have time to talk about it, but when we do a model of PTSD where we give an acute stress and then look 3 weeks later, or we cue that stress back up with a conditioned odor that we associated with the stress, it's a restraint stress, it will produce the exact same things I'm going to talk about in the brain that looks like virtually identical neurobiology that we would see when we use cue-induced relapse in our animal models. So they really do seem to share a neurobiology, but again, I don't really have time to go into the stress models. Okay, so this is where we've come to focus. And it's probably, I'm sure it's not the only place in the brain. I know it's not the only place in the brain that undergoes profound plasticity with drug use. But this is how we've come to think about it, especially as I would relate it back to that very anthropomorphic situation I just described between my buddy at the bar and my wife. So the brain has been reduced down to a prefrontal cortex, a nucleus accumbens, and the rest of it's been wrapped up into a box called the basal ganglia motor pattern generator. So the guy calls from the bar. That's I'll have a cortical understanding of what's going on. I'll draw on a lot of memories. I'll start pulling up how good the beer tastes, how fun it was to talk to everybody. That'll activate a set of neurons. In the animal models, we estimate that set of neurons in the cortical accumbens pathway there is probably 1% to 3% of the cells. At least those are the ones that show evidence of activating in response to a cue. And that would then lead off through the rest of the brain, and I'd head off to get a beer. When Sue calls me, there would be and I'll show you a little data indicating that they are largely separate ensembles from a natural reward versus a natural motivator versus a drug-related motivator. And if I wasn't an addict, I would go home. So what happens if it's an intrusive thought is you get the same thing. I start heading off to the bar, but something happens and the nucleus accumbens changes such that Sue, I certainly recognize that Sue's called me. I recognize it should be important, but it doesn't take on the motivational value that it should. And it basically gets outcompeted by the motivational value of the bar. And I continue to get a beer. So obviously, if we're going to treat that, we'll want to somehow take that yellow fog that I drew there, and you'll see it possibly is a gas, that yellow fog that I drew there, and you'd want to eliminate that so that at least Sue and the guy at the bar would have be on a level playing field and competing with each other. They both have completely functional ensembles. Okay, so here's our model of cue reactivity or craving. This is a mouse. You can see we have a nose ports. So when they push their nose into a port, more often than not, they get an infusion of, I believe this is cocaine in this particular example. The data that I'll show you can be recapitulated whether you're using cocaine, heroin, or nicotine are the ones we've studied the most. So there you can see the animals in the blue circles there learn the task really fast. They press the nose for cocaine. They don't go so much to the inactive nose port because it doesn't give them anything, and so they don't spend much time there. And then we extinguish that. So that basically we put them in the box. They can poke either nose port, and they don't get any drug. And so you can see over the course of a few days, they come back down and they hit what's called an extinction baseline. So hopefully you can see my cursor here. So this would be on an extinction baseline. They go in and they check the nose port. They still remember this is where they got the drug. But in fact, they just curl up and go to sleep. They don't perseverate at the nose poke because they've learned to suppress that behavior. I'll talk a little bit about the active process of suppression that's induced by extinction training and touch on the neurobiology there. We spent a bit of time working on that. But mostly what I'm going to talk about is what happens, what can we do to cause this animal that's in the box and it's suppressing its behavior. So it's extinguished its active nose port responding because there's not been drug for 2 weeks. So what can we do to cause this animal to jump up, even though there's no drug returned, and nose poke 40 or 50 times? So what we do is we have a cue that we've associated with each infusion here. So every time they got an intravenous infusion of drug, a light and a tone went off. So they didn't see the light and the tone here, but so that is not extinguished. And now we present the light and the tone, it's suddenly back. when the animal nose pokes in the active port, and they'll nose poke, 30, 40, 50 times, depending on the animal, just to get the cue. And so over the years, what we've done is we kill the animals either on that last day of extinction, 15 minutes into cue. We've done time courses, but the workhorse time is 15 minutes. And then we also have a yolk saline control. And then you'll notice down here, I list a sucrose control. And so all of the things I'll be describing to you, we have done sucrose controls on in a separate animal. Towards the end of my talk, I'll be describing how we do things in a dual model where animals they choose between sucrose and cocaine or heroin. And that's really where the future of our work is going. We've just published our first paper. It's in press in molecular psychiatry right now using that particular model. So that's the setup. And now we're going to go in and look at the synapses in the nucleus accumbens. So everybody's familiar with what a synapse is, and that the brain is a big place and it's very complicated. There's lots of connections. In the last 60 or 70 years, as neurobiology came into its own as a field, we realized that this is what neurons look like, and the synaptic connections, especially in the nucleus accumbens on the spiny cells, are on the ends of these spines that come off of the dendrites. And all the work, almost all the work, almost every paper, when you're trying to understand how cells are communicating and how they might change in a pathological condition, have been done there at the synapse. And that's cool. We've learned a ton of information, and a lot of the pharmacology that we have for intervening in psychiatric disorders, of course, is built on what we've learned about synaptic transmission and the receptors that are there, et cetera. But in fact, if you go into the brain, synapses occupy less than 1% of the brain. There's a lot of other stuff going on. Even if you look near a synapse, so that would be the synapse right there, there's other stuff right near it that could easily influence synaptic transmission. And we'll come back to what those other things are. But first, just a very quick primer on what we know happens at the classic synapse when an animal sees a drug cue and starts to press that lever or nose poke for the cue only. So no actual reward, just the cue. So this is a cartoon of a dendritic spine in the nucleus accumbens. You can see we put one receptor there, a beta-3 integrin. I'll talk about that briefly. And all of a sudden, the drug cue appears. And this is what happens, is using morphological measurements or measurements of synaptic strength, we can show that the spine heads get bigger within 15 minutes of this cue appearing, and that there's more AMPA receptors put into the spine. And then as you can imagine, if we run this assay for 2 hours, the mouse or the rat will start to realize, they're not as stupid as they look, they'll start to realize that the cue doesn't mean drug anymore. And their pressing will go by 45 minutes, depending on the animal, an hour certainly. Pressing is starting to come back down to baseline, and by 2 hours, it's down to baseline. So what happens to this, what we call transient synaptic potentiation? Well, transient is the key word there, because as the motivational value of the drug cue fades over 2 hours, the dendritic spine and the AMPA receptors come back to their baseline. So this is a transient event. It's very tightly associated with the motivational value of the cue. And as that value decreases in the course of a session, the plasticity wears off. So it's a different kind of plasticity than you're used to thinking about, where a synapse goes into long-term potentiation and that's the end of that, it stays there. This is a transient event, very much tied in with that behavioral event of seeking. And if it's a sucrose cue, exactly, that's what happens, nothing happens. We can't measure any change. I mean, maybe there's some change in a smaller population. I suspect there is, of neurons, but when we do our gross measurements at the level of all the medium spiny neurons, or even if we break it into D1s and D2s, which I'll come back to if that's not a familiar term to you, nothing seems to be happening with the sucrose cue. So the sucrose animal will nose poke like crazy for the cue, but this is not happening like it does when the animal nose pokes for drug. And just very quickly, those of you that are interested in this kind of thing, these two recent papers, Coty's done a stellar job of showing that this is how it expands. So there's this RGD, and we'll touch on that. But basically, it's an extracellular matrix interfaces with the beta-3 integrins on the spines, and results in the phosphorylation of focal adhesion kinase, and that causes the spine head expansion. The CAM kinase 2, we don't have as much evidence for. We have evidence for it, but it's not as well worked out in terms of the full signaling pathway. The bottom line is, if you knock down the beta-3 integrin receptors, you lose the transient synaptic potentiation, and the animals will continue to respond to sucrose cues, but not to the drug cues, whether it's heroin or cocaine. Okay, so that's a little bit of the signaling that goes on. We're not going to spend any more time on that. What I really want to jump up to is the fact that, so that's cool. We've got our endpoint here. It's the post-synapse, and we can show that it expands and it gets stronger and everything. And if you record, you can get ideas about what's happening in the presynapse in terms of release probability, and we can see changes there. But that's all you really have. The rest of the brain that I was showing you is generally treated by physiologists who study these kinds of things as dark matter. It's just kind of out there. It's a lot of it, but nobody knows exactly what its role is in synaptic transmission. And so, just like you can't really tell exactly what that picture was, it's our thesis that you really can't tell, or you're not going to fully understand and be able to manipulate something like transient synaptic plasticity if you don't understand what's happening in the other two compartments that are around the synapse. So one, most of you are probably more familiar with, which is astroglia. So astroglia send processes. Different brain areas have more astroglia right next to the synapse than other brain areas. In the striatum, it's probably close to 50% of the synapses are ensheathed in an astroglial process. And then you also have a proteinaceous matrix that lives outside the cell. It's been thought to be just kind of an infrastructure, just kind of how this glacier is an infrastructure in this picture. But in fact, it's a very active signaling domain. And we'll talk about how it signals. And in fact, that RGD is a ligand that comes off of the extracellular matrix to signal to the postsynapse. All right, so this is a real version of the tetrapartite synapse. And you can see the EM on the left, and then we made a cartoon out of it on the right, color-coded for the 4 compartments. I've already said everything I'm going to say about the pre and post synapse. So what we're going to talk about, and that was older data. We've got some newer data with astroglia. And so I'll spend a moment talking about that. And the first thing we had to do was figure out what do astroglia look like. And from the serial EMs, it was clear that they were going to make dendritic spines look like a piece of cake if we were going to quantify them. They're basically sponges is what their morphology looks like. And so you can sort of see that in this digital reconstruction of an astroglia that we filled with then digitally isolated, filled with a membrane reporter. And you can just see it's got all these processes going anywhere. And as I rotate it, you'll see little holes and things, and that's where dendrites are going through. And if it's a big one, it might be a small capillary or something going through the astroglia. And if we pull in close, you can see exactly what it is. It's just something that's wrapping around in the neural peel. So if you counterstain that for synapsin, which is a presynaptic biomarker, so it's only, you only find it in the presynapse. And then you look at where it overlaps. So this is not a FRET assay. It's cruder than that, but it's high resolution confocal microscopy. So if anything's within 200 nanometers of something else, it's going to look like they're on top of each other. So they're going to be what we call co-register. So if you stain for synapsin, all those purple dots are where the membrane of the astroglia is co-registering with the synapse. And basically we can just kind of go through and count those. It's a little more complicated than that, but we can see what's happening during transient synaptic potentiation, or when the animal is responding to the cue, what's happening to the relationship, the morphological relationship between the astroglia and the synapsin. And so the micrograph up here just shows basically what it really looks like in confocal. That would be synapsin in green. The red is the astroglia. We digitally isolate it, and then you can, these would be the purple dots here. So this came out over 2 papers. This shows the astroglia, and they have a pretty wide distribution in terms of, these have a reasonable amount of synaptic contact. These have almost no synaptic contact. During withdrawal, so this is 2 to 3 weeks after the last heroin session, these are heroin data, you can see that there fewer glia have significant contact with the synapse. Then after they get 15 minutes of cue, and you kill the animal and look, a subpopulation of those glia are now making contact with the synapse. But if you wait the full 120 minutes, when the animal has undergone what we would call within trial extinction, meaning the cue is no longer motivating them, everything's come back. So it parallels that synaptic plasticity that I was showing you, that transient synaptic plasticity. And during the cue, they go back to the synapse, or at least a subpopulation do. So we wanted to find out what that meant. Turns out that there's an actin binding protein called esrin. It's part of this ERM family that is only produced in astroglia, at least in adults, rodents, and it's localized out in the processes. So just for your edification, it was discovered in astroglia in culture, and one of the reasons culture work does have its issues is that's what an astroglia looks like in culture, which is nothing what it looks like in vivo. Nonetheless, you can see how the green, which is the esrin, is very much localized to peripheral processes. And so Anna got this way to knock down the esrin. And so you can see here it is, and the green dots this time are esrin, and there it is knocked down. And when it's knocked down, the astroglia no longer have as much co-localization with synapsin. So that's what it normally looks like, and it's been reduced because we've knocked down the esrin. And we can show that when they see the cue, that the phosphorylated state of esrin goes up as well, which I don't show you here. And so this, again, this is just showing that the esrin knockdown is knocking down the synaptic co-localization. It's not affecting surface GLT, and we'll come back to that. Okay, so if we knock it down, what happens to the relapse event? That's the main point. And it gets worse. So this is a control. AS refers to antisense. So this is a control antisense. And there's the relapse event. You can see most of it first 15 minutes, and it tapers off. And that's greater, significantly greater if you've knocked down the esrin. So it's actually a compensatory event. When the animal starts getting motivated by a cue, the glia, at least in some of the glia, go towards the synapses, and that appears to dampen the cue-induced reinstatement. All right, well, one of the things that the astroglia that are close to the synapse do is they express a protein called GLT1. So it's an astroglia specific, or at least 98, 99% of it's an astroglia. Glutamate uptake system. So it's a glutamate transporter that basically maintains fidelity of the glutamatergic synapse by after the glutamate is released here into the synaptic cleft. Oops, sorry. It'll interact obviously with the postsynaptic system, but then it gets taken up right away. So you don't get a significant amount of glutamate spillover out of the synapse. So it's maintaining a certain amount of synaptic efficiency. It's also preventing synapses from talking to each other, at least through this mechanism of glutamate spilling out. So what happens to GLT1? What you can see is, I don't show it here, but total GLT1 is knocked down. But what's on the surface isn't knocked down in an animal that's withdrawn or extinguished from cocaine. You can see at 15 minutes, again, as the animal's responding to the cue, GLT1 goes to the surface. So it's out there sucking up glutamate that's spilling out of the synapse. And by 120 minutes, it comes back just like everything else that's involved in this transient synaptic event. And if you look at the surface GLT1 relative to total GLT1, because as I mentioned, the total goes down, so it's normalized to total, you can see that there's a negative correlation. In other words, the more GLT1 that goes to the surface, the less they reinstate, the less they respond to the cue. The less GLT1 on the surface, the more they respond to the cue. So again, this looks like a compensatory event. So glia are actually trying to dampen the behavioral response by putting GLT1 on the surface to suck up glutamate that's spilling out of the synapse, and by putting processes and GLT1 a little bit closer to the synapse. So the other fourth component that I want to talk about briefly is the extracellular matrix. And you can see that here in this, you blow up this EM, you can see that there are proteins out there in the presynapse, postsynapse. You can see these extracellular proteins. I mentioned it's a signaling domain. So proteins, how they signal is they get cut. So it's a catalytic signaling. So there are enzymes out there called matrix metalloproteinases. And the two that we've studied and that most people have studied in the brain, they're in pretty high abundance along with MMP3, are gelatinases, which means they cut gelatin motifs in proteins. And that's a very common motif in the extracellular matrix. Fibronectin, for example, has it. And there are two MMPs that are gelatinases. One is constitutively expressed. In other words, it doesn't tend to respond that much in an acute manner to synaptic activity. The other, MMP9, tends to can increase its activity in response to increased synaptic activity. In fact, if you inhibit MMP9, you can prevent things like classic forms of synaptic potentiation, like long-term potentiation in the hippocampus. So some work about 10 years ago showed that, wow, these things are probably pretty important in synaptic plasticity. So Alex basically, and then Vivian came along later, and I'll show you her data next, showed that they were important in this event that we're talking about and in the cue-induced reinstatement, again, to drug, but not to sucrose. So how we measure this is we put in a false substrate. So it's basically we microinject some gelatin into the nucleus accumbens that is quenched. So it's got a FITC, a fluorescent molecule, quenched inside of the gelatin. And as the gelatin gets digested by active enzyme, one of these two MMPs, it'll start to fluoresce. We mask out the injection site here so that the dotted lines here, you can see a little fluorescence in there. It's because just the damage from the injection will produce some activation of MMPs. And this is the anterior commissure, so we know that we're in the nucleus accumbens. And you can see even in cocaine extinction, not in heroin, but there's MMP2, it turns out, activation that's kind of constitutive. So 2 or 3 weeks after the last cocaine self-administration, you have activity. So this is just constitutive activity that's gone up in the cocaine brain. Again, heroin did not do that. Nicotine looks like it did. But the real take-home message for our purposes today is that when the animal reinstates, so when they start responding for that cue, signaling takes off in the extracellular matrix, and it is MMP9. If we give an MMP9 inhibitor, we prevent this, and we prevent the animal from responding to the cue. And you can see that if you wait for 120 minutes and look at it, it comes back down. So it's just like the other transient synaptic activity. So not only is the pre and post synapse flexing in response to these cues and the astroglia, but the extracellular matrix is as well, which isn't surprising when you consider that that's all this morphological activity and it's embedded in an extracellular matrix. But this shows that it's happening, and it shows that it's needed for the relapse event to occur. Not for sucrose. You could relapse to sucrose cue just fine if you block these MMPs, but you cannot respond to a heroin cue or cocaine cue. And Vivian's gone in a little more closely. She's now back in medical school. She's an MD PhD student. Did a great job of taking this fluorescence down to a cellular level. And so you can see it's still green, and this is filled dendrite. And then she can quantify the different states. So this is the saline animal. This is the extinguished animal. This is the reinstated animal. So 15 minutes of cue. And if it's a D1 medium spiny neuron, which is half the neurons in the nucleus accumbens express D1 receptors, the other half express D2 receptors selectively, and about 5% don't express either one. Okay, what you can see is MMP activity goes up during the reinstatement event. And again, this can be inhibited by MMP9. In the D2 neurons, it goes up during the extinction, which means that it's a constitutive change. So you've got this constitutive elevation. And then when they see the cue and they start responding, it actually has to come down. So I don't have time to go into her whole study where she showed the mechanism by which this comes down and things like that. But it's very cool that this one cell type shows activation. So the matrix is getting activated transiently on the D1s. It's actually being turned off transiently by a cue on the D2s. And as it turns out, the transient plasticity is occurring on the D2s is more related to the extinction event, whereas the D1s is more related to the seeking event. I just don't have a lot of time to go into that. So here's the overall story that we've put together. If the guy in the bar calls, you get a release of glutamate, and that hits the postsynaptic system. But as you recall, the glia have pulled back. That's the resting state. And GLT1 is down-regulated. So glutamate spills over. GLT1 responds to that spilled over glutamate as best it can, as we showed with the glia coming back and bringing GLT1 back. But there's a big spillover event. I don't show you the data, but that's been measured by us and people, many people. So there's a spillover event as the animal's responding to the cue. And in publications I don't have a chance to show you, it acts on interneurons that produce nitric oxide. And this is why we made that little yellow thing in the original cartoon, kind of a putrid gas that goes out. Well, that's nitric oxide. It's a gas. It nitrosylates MMP9, which activates it, cuts the matrix, interacts with the integrin and potentiates the synapse. If it's a sucrose cue or, in this case, the anthropomorphization, a spouse cue, basically, I do the adaptive behavior. I get in my car and drive, but this is a very well-regulated synapse. The glia are there, the GLT1 is there, and the glutamate gets taken up, and you don't activate the rest of the system. If anybody's interested in those papers, I can just email me. I'm more than happy to send them to you. So I don't have too much time. This is where we're headed with this sort of trying to really look at the ensembles and getting at this the way I've set this talk up, really, which is anthropomorphically, where you have a biological cue competing with the drug cue. So if you assume they're separate engrams, so the drug cues are the red ones and the spouse cues are the green ones. When you have TSP, so now you're expanding a bunch of the neurons, you're potentiating a bunch of the neurons in the nucleus accumbens. That would be the yellow ones. And we know it's a lot of them. Turns out it's mostly D1s, MSNs, but it's a lot of them because we can measure it very easily by just blindly patching onto the cells. Then when I get the call from my spouse, or the spouse is called first, but then the drug produces this TSP, it kind of swamps out. This is the theory. It swamps out the biological cue such that the animal's motivation is oriented towards the action plan that is related to the drug cue, not the biological cue. So that's the global hypothesis of what we think is going on and what we're trying to set up to study very clearly. And so I just want to show you very quickly, some of the things. This is AC's, Anna Clara's paper that's impressed. She's now just took a job at the University of Wyoming. She's from Paris, so it'll be an interesting thing to see what happens in Laramie. But this is what she set up. So you have a sucrose pellet or a cocaine infusion. So alternate days, and you can see, so that's the sucrose active response. That's the cocaine active response. This is the inactive. You can already see that something's happening here, that they perseverate on the cocaine nose poke, which would be the inactive, this is the sucrose day, which would be the inactive nose poke. So they kind of stay on the cocaine one a little bit longer than when they go to a coke day, and they don't stay as long on the inactive, and they just go straight for the coke one. But the bottom line is we can titrate everything so that they're nose poking about the same, whether it's a sucrose day or a cocaine day. And then we extinguish them. And here's the take home message right here, is that if we bring both cues back, but they're getting a reward, they go for the sucrose. So if the rewards are available, and other people have shown this, we weren't the first people to show this particular event, they actually prefer the biological reward if both rewards are available. But what about out here, once they're extinguished, and now they're just competing in a more existential way? In other words, the cues are competing for each other, not the actual rewards. And what you find is that the drug reward dominates. So they will spend more time at the drug nose poke than with the sucrose nose poking. So this is what we want to find out. We believe this is the transient synaptic potentiation. We know that there's no transient synaptic potentiation on reward delivery. In fact, if they get the reward, it will inhibit. If they get the drug, comes back, it inhibits the transient synaptic potentiation. So we think that the reason the cues are so much stronger for the drug than the biological reward is that transient synaptic potentiation. This is a duplicate slide. So AC set up this FOSS trap model. I don't really have time to go into the model, except to say that when you open up the transgene, it will then, during that time, anytime that FOS goes on, which is a immediate early gene that indicates the cell's been activated, not 100% pure indicator, but that's generally the gist of it. Instead of just going up and going away, it will permanently deposit a reporter. In this case, it's a D tomato reporter, so it looks red. So then you can go in and you can see the cells that are responding to that cue if you use a cue to activate it. And so that's what the case is here. So, excuse me, what you can do is then come back and at a later date, show the cue again, or show the cue for the sucrose, for example, if the red was for the cocaine, and see if the green ones, which are for the sucrose, and in this case you have to kill the animal because it's just a straight up protein measurement for the CFOS, overlap with those from the cocaine. This allows us to tell exactly how many of the sucrose cells are the same as the cocaine cells in responding to the cue. So what we find is that there's this is just summarizes her findings. There's about a 30% overlap. The seeking ensemble, so you can also get an ensemble for the extinction event. The seeking ensemble is about 72% D1s, whether it's a sucrose or a coke ensemble. And the extinction ensemble either way is about 64% D2s. So this sort of D1, D2 organization is holding up. I'm just going to go on here to this. So very quickly, and then I'll come to the clinical trial. We've started to do a lot of calcium imaging, and this Retta's doing a really great job of this, of the D1s versus the D2s, to try to watch the ensembles together. We're just starting to do it where we actually do that more complicated paradigm, so we can watch this, actually tag or visualize online the sucrose ensemble versus the cocaine ensemble. But I'm pretty much out of time. Without going into great detail, I'm just gonna, this has got most of the information, it's just not as elegant. If you look at the D1s, what you can see is this is the seeking event here. And you can see the D1s are more active overall. And when they're extinguished, so now they're extinguished, the D1s become less active. So these are the same cells in two different tests, the relapse test and the extinction test. You can see it's the opposite for the D2s. Overall, the D2s are more active in extinction than they are the D1s. And sorry, I don't have time to really go into the rest of this cool stuff. This is just, you can ask questions if you want to understand why we think this, but we basically think what's happening is during extinction, the D2s are showing more tonic activity and they're suppressing the D1s. And that even during the course, which is what I didn't have time to show you, the course of a relapse event where they start to show within trial extinction, in other words, they're not interested in the cue anymore, these become more active and they suppress the activity of that. And that's what all the pretty colors were showing, is that that's what's happening. And so now we're going in and we're going to inhibit one or the other and try to show how they're related. Okay, so what does all this mean for drug treatment? Very quickly, there's a lot of targets that are in this. The one we hit on was one of the first ones we discovered, which is bringing GLT1 back. Let's bring GLT1 back, soak up the glutamate, and maybe that'll turn a coke relapser into a normal relapser. So these are the two people I think that made the biggest contribution to the N-acetylcysteine story. One is Dave Baker, who sort of stumbled on it in the very beginning, and then Lori proved that he was right in principle, but wrong in detail. And so that came out here. So Dave basically focused in on the cysteine glutamate exchanger as the target, which it is. But Lori showed that the real action with N-acetylcysteine is to restore GLT1, even though it also restored the cysteine glutamate exchanger. But the restoration of GLT1 appears to be the active ingredient in suppressing relapse. So you can see, if you look at the dates, that was 2010. We actually started a clinical trial thinking about the wrong target, the wrong molecular target. But NAC was already being used clinically, so we were able to put it into a Q reactivity trial, and a very simple little kind of phase 3 essentially is what it was. And the bottom line is that if a person came back on alternate weeks, either getting NAC or placebo, they were shown either a drug Q or a neutral Q. They were allowed to switch off the slides if they were bored with them, or would switch automatically in 20 seconds. This is the amount of time they would stay on the slides. And so obviously they spent more time in the control situation, and these were Coke users, control situation on the Coke queue than the neutral queue. But when they were on the NAC, they spent less time, still spent more time, but less time on the Coke queue than they did in placebo. So this was taken to be the same thing as giving NAC in an animal didn't press as much for the drug queue, which is what we'd shown over and over again, and others now have in animal models. So this led to a big full-on trial, and I'm just going to give you the results. No effect on relapse rates. Still suppressed craving, but didn't affect relapse. But if we isolated those that went into the study abstinent, it had a good effect on relapse. So those of you that are clinicians in particular are used to looking at these survival curves. And so these are the NAC-treated patients, and they were on NAC for 8 weeks. And you can see, regardless of the dose, some of the individuals that started off abstinent never did relapse. So when they all relapsed, they're down here, the curve is down here. And you can see those that were on placebo, the majority of them relapsed right away within the first half of the trial. And so it looked like if they were abstinent, it might work. So this has led to things like, let's try it on smokers where they're on varenicline to make them abstinent, and then put them on the NAC to try to sustain the abstinence. And the results are not in from those trials. But what it caused Sudi and I to do, we got this DoD grant. Actually, it came from the Gallo. They used to manage a large DoD grant. I don't know who's managing that now, or if it even exists at UCSF. But they used to distribute funds and for the DoD, and we got a grant to do this small clinical trial. So I was basically just the cheerleader. Sudi did the trial, and we wanted to do it in vets so we could get people that were abstinent. So we know they were abstinent going into it. And there's kind of a halfway house environment that would sustain their abstinence, but they were also comorbid for PTSD. So this is basically before they start the NAC. This is week 4 of NAC, week 8 of NAC, and then 4 weeks after discontinuing the NAC. On week 12. So you can see, like it always is done, we've shown this, and many people have shown this now, that NAC suppresses craving. So it does that, or as we would call it, intrusive thinking, if you wanted to use the broader term. The big surprise from this particular study was that this CAPS questionnaire, if you're over 50, you're diagnostic for PTSD. And so what you can see is that it brought everybody below diagnostic criteria for PTSD. And one of the big endophenotypes of PTSD, of course, as I mentioned, is intrusive thinking. And it really shut down the measure of intrusive thinking. And so this is really the study, you can see it was done in 2017, that got us to think about that maybe what NAC is working on, because there were also trials now with rumination we didn't do, Mike Burke in Australia did these in depression. There have been some successful trials with trichotillomania, a form of OCD, hair pulling. And so we started really thinking that it's not an anti-addiction drug. It's just working on one of the symptoms of addiction, which is craving, which may or may not be enough to stop somebody from relapsing. Okay, and this is just a slide that shows all of the trials for different things. The ones in orange are the ones that failed. All right, so I've always been very lucky to have a great lab. I highlighted with pictures some of the people that were really important in the data that I showed you. One of the things about stepping down from chair, I'm no longer chair of the department, I stepped down a year ago, is that I can't control the Halloween party. And so we don't have a departmental Halloween party anymore. And we had to go to a pub down the road to have our Halloween party this last year. I don't know what we're going to do this weekend, probably nothing. We have a center here and these are the people in the center. There's no way we can get anywhere near where we've gotten with some of these weird ideas if we didn't get support and encouragement from these folks. And I've been fortunate with funding over the years, and so especially from NIDA, very grateful for that. Thank you very much. I'm sorry, it looks like I used up my hour and then some.
Thank you so much, Peter. That was really great. We do have a couple of questions in the Q&A, and if anyone wants to add questions, please go ahead. I'll just read those out to you.
Okay.
Howard Fields asks, what are the major side effects of NAC? Does it include sedation?
Yeah, so there's nothing, basically. The only thing that ever comes up above placebo is nausea. And that's really the formulation. So basically it's got disulfide bonds in it. And so it stinks. It smells like rotten eggs. So we make a cap that theoretically doesn't dissolve until it gets pretty deep into your digestive system, and that helps control the nausea. But if you take high enough doses, like when you get up over 3 grams, you start seeing a significant number of people will complain that they're feeling a little nauseous when they take it, and they have to take a bucket of this stuff. Dave actually went off to start a company called Promentis, and they now have in clinical trials a NAC-like compound that you only have to take, instead of taking up to 3 grams a day, you take, the trial's about 100 milligrams, and it's in trial for smoking and trichotillomania, and it doesn't produce any of the nausea. So I think the future holds a non-nauseous version, but there's literally, there's no other side effects.
Howard also asks, it looks like there's a certain amount of overlap of the effects on D1 versus D2 neurons in the nucleus accumbens. You said this is all in the core, yet there's some evidence, including some from your lab, that the infralimbic projection to the shell is at least as involved in extinction. Do you have any shell nucleus accumbens medium spiny neuron data you'd like to tell us about?
Howard's always a troublemaker. No, thanks for the question, Howard. No, we don't. So we don't do much in the shell. We have one study where we found, for example, we put an animal into a extinguished environment and then kill them at 120 minutes, at 15 minutes or 120 minutes, make slices and look at AMP and NMDA ratios, or look at spine head diameter. The spine head diameter doesn't change, but the AMP and NMDA ratio goes up in the D1, in the D2s, not the D1s. So this is the animal that's actively extinguishing. They've already learned the extinction event. And that was all done in the core because that's where we do everything. So as part of Doug's thesis, I had him go back and say, well, we got to see what happens in the shell for the very reason that Howard pointed out, that we've earlier published papers about this connection between the infralimbic and the shell and its involvement in extinction. And it seems to do the same thing in the shell, but it actually is a more profound effect in terms of this form of extinction-induced plasticity in the core than it was in the shell. So that's really the only shell data that we generated on purpose anyway.
Okay. Just building on that for a moment, in your anthropomorphic cartoon, you do sort of suggest that there's a difference between the prelimbic cells and the infralimbic cells. Is that what you were kind of alluding to in the drawing, or were
No,
those different, just the
no,
red and
not,
green that could be intermixed in a particular part of cortex?
yeah. I mean, you're very familiar with, I'm sure, the difference in the projections coming out, and the topography. And so the infralimbic definitely goes to the core, but it's biased towards the shell and vice versa with the prelimbic. So we assume that there's something really important going on with that. And with cocaine, it's very clear, at least in our hands, and I think, it's been replicated, but it remains a little bit controversial, the infralimbic part. But the prelimbic definitely drives drug seeking. So I think everybody shows that with all different drugs. And that's mostly an output to the core, although it does go to the shell. The infralimbic, there's plenty of data, more in the fear conditioning literature, that it drives extinction of learned responses, the output to the shell. But there's also, there's scattered papers that show that it's also important in seeking. And so it's not a pure event there. And I think we have to go in with some of these calcium imaging, where you're doing specific cell types, maybe that have been retrogradely labeled from the shell versus the core, and see if one or the other type of projection is more or less responsive to either the extinguished environment or the drug cue. So I think we can't answer that. In terms of my art, I'm just a crummy artist. I have no intention of trying to distinguish the two.
Fair enough.
It's just prefrontal cortex.
And we do have one more question from Alan Bassbaum. Asking about comorbidity of drug dependence and depression. I guess this is in the context of your NAC effects. Has depression worked into your clinical trials or anything that people have done with NAC at all?
No, not that I know of. The person who's done almost all of the work with depression and NAC is Michael Burke in Australia. He's got a number of papers, half dozen papers probably at different trials. Somewhere in there may have been something done, or at least, they didn't have drug addiction as an exclusion criterion. And so they might have been able to comment at least on people that were also drinkers or also smokers or something like that. But I don't know if that's in there. Again, Michael Burke, if you look him up as Berke, you and kind of look through his papers, maybe you'll find something in there. That would be the only place I can think of that might have something about that particular comorbidity.
And this is also sort of coming out of a question from Alan related to ketamine. And so the NAC target is related to glutamate, right? Probably. So the first part of the question was, is there any evidence that ketamine is efficacious? I guess in the same models as you're looking at for NAC, or that people have looked at for NAC. And do you have a hypothesis about how ketamine functions as an antidepressant?
In theory, that I've mentioned it very briefly in passing about varenicline, which is known to produce abstinence in smokers. And then maybe you'd add in NAC afterwards, and that would help sustain the abstinence, because with varenicline, I forget what the number is. I think 75% relapse by 6 months, something like that, if they're just on the varenicline. So maybe NAC could push that out a little bit. So in theory, as I understand the use of ketamine in depression, and again, I'm not the clinician, in theory, you could do the same kind of thing. You could knock them out of their depressive state with the ketamine. And then if you prevented rumination, which in theory, the NAC should do, because it's working on that same mechanism, they might not become depressed quite so quickly again. They may not relapse to their state of depression. So that would be my suggestion. When ketamine first came out as an antidepressant, we poked around with it a little bit in this model. This would be many years ago. We only advanced it as far as a few little preliminary behavioral trials. There was not anything to write home about. I haven't really kept up on that kind of sort of behavioral pharmacology literature. So there may be something out there that if you did a search on, ketamine and cued reinstatement or something like that, maybe you would find that there was a positive study, but we, in a very quick, relatively quick analysis, didn't demonstrate anything dramatic, at least.
Mm-hmm. Okay, well, we are a little over our time and there are no more questions in the Q&A. So again, Peter, thank you so much for being willing to give this virtual talk for us today. And hopefully next time we'll be able to catch up in person.
Yeah, no, it'd be great to do it. We're all looking forward to the end of this thing so we can get together. Thank you very much. It was very, very helpful for me, as you might have gathered. A lot of that calcium imaging, it's the first time since nobody's running around giving talks anymore. So it gave me a venue to put that together. And so, it's very useful for me, and I hope it was for other people as well. Okay. Thank you very much.
Great.
Goodbye from Charleston.
Bye.
See you.
Bye. Thank you.
Yeah.